Good afternoon, girls. Good afternoon, sir. Tirupati sir. Welcome, girls. Today, student activity program. This one is the student activity program. Our students, Reddy College students, to present the PPTs. GDC College, Jagitial Government Degree College, Jagitial students. Same, both students are second year, fourth semester syllabus. My students. Okay, girls. Sir, how many students joined your students?
Yes,
Tirupati
madam. My
sir.
students also joined. They will already join, and some more students will join.
Student, to start the presentation, ma. Girls, share your screen and to start the presentation.
Yes.
Yes.
Meeting, ma'am.
Once to check, ma. Usha.
Yes, madam.
Good afternoon.
Good afternoon. Myself, Neha. It is a group project. Our topic is cytokinesis. Cytokinesis in plant cell which have cell wall. Cytokinesis occur by a process called formation of cell plate. Cell wall material collect in the cell plate as it grows. The cell plate enlarges until its surrounding membrane fuses with the plasma membrane along the perimeter of the cell. Two daughter cells result each with its own plasma membrane. A new cell wall arising from the content of the cell plate is formed between the daughter cells. What are the cytokinesis? Cytokinesis are low molecular weight regularity protein or glycoprotein secreted by the white blood cells and various other cells in the body in response to a number of stimuli. They play major role in the induction and regulation of the cellular interaction involving cells of the immune, inflammatory and hematopoietic system. Thank you, ma'am. My friend Manisha will continue.
Good afternoon. Types of cytokines, lymphokines, cytokines secreted by lymphocytes, monokinins, cytokines secreted by monocytes and macrophages, interleukins, cytokines secreted by some leukocytes and act upon other leukocytes, interferons, cytokines secreted in response to viral infections and other stimuli, tumor necrosis factors. They are secreted by some macrophages and they induce the necrosis of tumor cells and pro-inflammatory actions. Chemokines. They low molecular weight cytokines that affect chemotaxis and other aspects of leukocytes behavior. First, lymphocytes. Mixed group of proteins. They are not identified chemically, classified based on biological activity. Cause aggregation of macrophages at site of infection, chemotaxis, activate macrophages to phagotose. End result is amplification of inflammatory response and recruitment of immune cells to cell site. Next, monokine. Monokine is a type of cytokine produced primarily by monocytes and macrophages. Example include interleukin 1 and tumor necrosis factor alpha. Other monokines include alpha and beta interferons and colony stimulating factors.
Unable to hear you, girls.
Good afternoon, everyone. This is Akila. Interleukins, secreted proteins that bind to their specific receptors and play a role in the communication among leukocytes, assigned to each family based on sequence homology and receptor chain similarities or functional properties. Actions, classifications, type 1 cytokines, type 2 cytokines. What are interferons? Interferons are made by cells in response to an appropriate stimulus and are released into the surrounding medium. They then bind to receptors on target cells and include induced transcriptions of approximately 20 to 30 genes in the target cells, and this results in the antiviral state in the target cells. Thank you. Now remaining will continue by Bindush.
Good afternoon, ma'am. Myself, P. Bindustri. Chemokinesis. Chemokinins are small heparin binding proteins that direct leukocytes to the site of inflammation and injury. Chemokines are 8 to 10 proteins with 20 to 70% of homology in amino acid sequence. Chemokines act like chemo-attracted cytokine. Functions of cytokines. Activating signals for cells, activate, regulate cell cycle, meiotic activity, cause changes of the cell membranes, increases of cytokine receptors excretion, participate in respiration of tissue in the terminal phase of inflammation, regulate immune cells and differentiation in the immune organs, influence migration of the immune cells. Thank you, ma'am.
Is my screen visible, ma'am?
Yes, ma.
Okay, ma'am. Good afternoon, everyone. I'm Sahiti from FNBC Group Second Year, and our topic is abscisic acid. This PPT was prepared by Madhuri, Ashita, Vaishnavi, and Nivita. And now Jahnavi, Prasanna, Gayatri, and I are going to explain about abscisic acid. Abscisic acid. Abscisic acid is a plant hormone. ABA functions in many plant developmental processes, including seed and bud dormancy, the control of organ size and stomatal closure. It is especially important for plants in the response of environmental stresses, including drought, soil salinity, cold tolerance, freezing tolerance, heat stress, and heavy metal ion tolerance. During stress conditions in order to cope up with the environmental changes. So it is very helpful for the plant in that way. Functions of abscisic acid. ABA was originally believed to be involved in abscission. Abscission means the detachment of plant parts naturally, typically dead leaves. So earlier, abscisic acid was known only to be involved in abscission. However, it is not the case. This is now known to be the case only in small number of plants. ABA-mediated signaling also plays an important role in plant responses to environmental stress and plant pathogens. The plant genes for ABA biosynthesis and sequence of the pathway has been elucidated. ABA is also produced by some plant pathogenic fungi via a biosynthetic route different from ABA biosynthesis in plants. In preparation for winter, ABA is produced in terminal buds. This slows plants' growth and directs leaf primordia to develop scales to protect the dormant buds during the cold season. As we discussed earlier, that abscisic acid helps in cold tolerance. So during winters, as it is very cold, abscisic acid is produced in terminal buds, which helps in protecting the dormant buds from the cold weather. ABA also inhibits the division of cells in the vascular cambium, adjusting to the cold conditions in the winter by suspending primary and secondary growth. So by suspending the growth, the abscisic acid is protecting the plant from any damage during the winter. Now my friend Jahnavi is going to continue with the functions of abscisic acid. Thank you.
Thank you, Saiti. Good afternoon, everyone. I am Jahnavi from FNBC second year. Abscisic acid is also produced in the roots in response to decreased soil water potential and other situations in which the plant may be under stress. ABA then translocates to the leaf, where it is rapidly alters the osmotic potential of stomatal guard cells, causing them to shrink and stomata to close. The ABA-induced stomatal closure reduces transpiration, thus preventing further water loss from the leaf in times of low water availability. A close linear correlation was found between the ABA content of the leaf and their conductance on a leaf area basis. Saiti, next slide, please. Saiti, next slide, please.
Is it visible now?
Sorry, ma'am.
What happened, Ma? Continue.
Seed germination is induced by ABA in antagonism with gibberellin. ABA also prevents loss of seed dormancy. Several ABA mutant Arabidopsis thaliana plants have been identified and are available from the Nottingham Arabidopsis Stock Center. Both those deficient in ABA production and those with altered sensitive to its action. Plants that are hypersensitive or insensitive to ABA show phenotypes in seed dormancy, germination, stomatal regulation, and some mutants show stunted growth and brown yellow leaves. These mutants reflect the importance of ABA in seed germination and early embryo development. Pyrabactin is a naphthalene sulfonamide hypocotyl cell expansion inhibitor, which is an agonist of the seed ABA signaling pathway. It is the first agonist of the ABA pathway that is not structurally related to ABA. Now my friend Gayatri will continue.
Thank you, Janavi. Good afternoon, everyone. Myself, Gayatri from FNBC second year. Abscisic acid is an isoprenoid plant hormone which is synthesized in the plastridiol 2-methyl D-erythroyl 4-phosphate MEP pathway. Unlike the structurally related sequiterpenes which are formed from the mevalonic acid, derived precursor farnesyl diphosphate, that is FDP, the C15 backbone of ABA is formed after cleavage of C40 carotenoids in MEP. Zeaxanthin is the first committed ABA precursor. A series of enzyme catalyzed apodeoxygenations and isomerizations via violoxanthin, and final cleavage of the C4D carotenoid by a deoxygenation reaction yields the proximal ABA precursor, xanthozine, which is the further oxidized to ABA via abscisic aldehyde. Abamine has been designed and synthesized, developed, and then patented as a first specific ABA biosynthesis inhibitor, which makes it possible to regulate endogenous level of ABA. Sahiti, next slide, please. Coming to locating and the timing of ABA biosynthesis. Synthesized in nearly all plant tissues, example like roots, flowers, leaves, and stem. Stored in mesophyll cells where it is conjugated to glucose via uridine diphosphate glucose transferase, resulting in the inactivated form of ABA glucose ester, activated and released from the chlorenchyma in response to environmental stress such as heat stress, water stress, salt stress. Released during desiccation of the vegetative tissues and when roots encounter soil compaction. Synthesized in green fruits at the beginning of the winter period, and synthesized in maturing seeds, establishing dormancy. Mobiling with the leaf and can be rapidly translocated from the leaves to the roots in the phloem. Accumulation in the root modifies lateral root development, improving the stress response. ABA is synthesized in almost all cells that contains chloroplast or amyloplast. Remaining topic is continued by my friend Prasanna. Thank you.
Thank you, Gayatri. Good afternoon to everyone. My name is Prasanna Kumari from FNBC second year. Respected sir and madam. Topic is abscisic acid. Inactivation of abscisic acid. Abscisic acid is also inactivated by conjugation with glucose to form abscisic acid glucosyl ester. It can be catabolized to phasic acid cytochrome 707A. A group of cytochrome 450 enzyme are inactivated by glucose conjugation. The enzyme is uridine diphosphate glucosyl transferase. Cytochrome enzyme 707 enzyme is very important for abscisic homeostasis. Generally, it accumulates higher levels of abscisic acid that lines are overexpressing of biosynthetic genes. In soil bacteria, it is an alternative catabolic pathway leading to vermifolial. The enzyme is vermifolial dehydrogenase has been reported. Sahiti, next slide. Next slide, the effects of abscisic acid. Abscisic acid acts as growth inhibitor and induces bud dormancy in a variety of plants. Abscisic acid is a powerful growth inhibitor. Antitranspirants induce stomatal closure, decreasing transpiration to prevent water loss. It inhibits fruit ripening. It is responsible for seed dormancy by inhibiting cell growth, inhibits seed germination. It inhibits synthesis of kinetin, nucleotide. It down regulates enzyme needed for photosynthesis. Next, so it slow growth, seed dormancy. Abscisic acid helps suppress growth of dormant birds. Abscisic acid closes stomata. Abscisic acid promotes sleep sequences. And dormancy it used to induce dormancy in the seeds. It's physiological effects of abscisic acid. It used as anti-transpirant in drought-prone areas. Water stress is serious problem in agriculture production. So sprays of abscisic acid. Abscisic acids are suggested that gives partial closure of stomata for few days. It reduces transpirational loss of water. Here some important point: developmental and physiological effect of abscisic acid plays important primary regulator roles in the instation and maintenance of seed and bird dormancy, and in plant response to stress. Last in my conclusion of abscisic acid is the main hormone involved in seed dormancy. Seed dormancy is necessary to ensure the survival of seeding and to maximum seed dispersal. Thank you, ma'am. Thank you for giving me this wonderful opportunity, ma'am.
Girls, next ma. Girls, who is the next?
Ma'am, completed, ma'am, 2 groups.
Okay. 2 groups are completed?
Yes, ma'am, completed, ma'am.
Yeah. GDC girls, if any doubt to ask me, ma. Our students to clarify your doubts. Girls. If any doubt to ask girls, తెలుగులో అయినా అడగచ్చు. ఉన్నారా అమ్మ మీరు? Okay, Usha, end the session, ma.
Okay, ma'am. Thank you, ma'am.
Thank you, girls.
Thank you,
ma'am.
girls. Thank you, ma'am.